In this video, we will discuss about the different mechanism of targeted protein degradation. Though the basic idea will remain same, that they will use the natural protein recycling process within the cells to degrade their targeted proteins. So in the first case, this approach is suitable for the soluble protein also, or the soluble protein of interest also. So what they do is they make a sweeping antibody. So on their Fc side, it can interact with the soluble protein of interest, and on the back side, it can interact with this FcRn or Fc gamma R2b. And this, once it interacts to this FcRn on FcR gamma R2b, what it will does is it will recycle or degrade the targeted protein using lysosomal degradations. So first what it will does is it will go to this sorting endosomes, and where from there it will go to the lysosome degradations through this pH adjustment. And after that, and during this lysosomal degradation, what will happen? Only the protein of interest will be degraded, whereas the receptors will get recycled back. And in the other approach, it is based on the glycan based approach for the targeted protein degradations. What it does is they will combine all the ligands into the single molecules, which they call as MODE, and it can interact with the soluble protein of interest through this ligand glycan interactions. And once it interacted, it can the other part of the molecules will interact with the cell surface receptors. This will helps in the internalizations of this targeted protein, and that will leads to the lysosomal degradations. And another form of this MODE is this LYTAC, that is lysosomal associated chimera. So it does the same works, that is LYTAC interact with the cell membrane, and the Fc region will interact with the membrane protein of interest. It will also does the internalizations, and from this sorting endosomes, it will degrade the proteins and recycle back the receptors. Afterwards, another mechanisms that they talk about is to exploit the E3 ligase that is present on the transmembrane region. So here they make this PROTAC or ABTAC. In one arm of this, the PROTAC will attach to the targeted protein, and another arm will attach to the E3 ligase, and this will cause the ubiquitinations of the targeted protein, and thereafter it will leads to the internalizations of the internal part of this targeted protein. And it could also be a membrane targeted protein or membrane protein of interest, and this will then follow the same procedure, that it will go through the endosomal sorting. From that, all the receptors will recycle back, and the targeted protein will goes to the lysosomal degradations or proteasomal degradations. The next one is they utilize the natural cytokine that is recycled. So in this case, the ChITAC that is made has the natural cytokine, natural recycling cytokines antibodies or receptors, natural recycling cytokines receptors at one end, and at other end it will bind to the target protein. It can also work on the soluble protein and on the membrane protein of interest. And in that case, once it is recycled, then this endosomal sorting occurs using the change in the pH, and this will thereafter direct the targeted protein towards lysosomal degradations and recycling back of the natural cytokines. And afterwards, in the next mechanisms, here they exploit the use of the recycling integrins. So here they use the antibodies that are designed in such a way that at one end it will interact with the recycling integrins, and at other end it will interact with the targeted protein of interest. It can also work for soluble protein and the membrane protein of interest. So this is basically a bifunctional small molecules, but one end is specific to the targeted protein and another end is specific to the recycling integrins. And thereafter, through this endosomal sorting, it will direct the protein of interest towards lysosomal degradation and recycling back of the integrin receptors. Last one is the one that we discussed in the previous video. It is PROTAC. PROTAC is a small molecule where one end is attached to the protein of interest. It could be membrane protein of interest or the soluble. Soluble region is not shown here, but one end is attached to the membrane protein of interest and another end is attached to the ligase, ubiquitin ligase. This will lead to the polymerizations, E3 ubiquitin ligase. This will lead to the polymerizations of the membrane protein of interest through this ubiquitin by addition of multiple ubiquitin, and this multiple ubiquitin will then direct them towards proteasomal degradations that will lead to the degradation of the protein of interest that are being targeted. So this is how the different mechanisms works, although basic idea is same, that is to use the natural protein recycling process for targeted protein degradations. I hope you understood different mechanisms to some extent. If you need to understand more about this mechanisms in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.